Welcome to AP Biology 2.6 Membrane Transport. This is Dr. Joseph. In this chapter, we are going to study the different forms of transport. Specifically, we're going to look at the difference between passive versus active transport. And in passive transport, we are going to look at the different types of diffusion. And in active transport, again, we are going to see the two different types of active transport. And we're also going to explore the different types of proteins involved in transport across cell membranes. So, as we saw, our cell membrane is a semi-permeable or a selectively permeable membrane. It allows some things to pass through and other things to not pass through. So, if the molecule is small, like carbon dioxide or oxygen and not charged, small uncharged particles can freely diffuse across the membrane. Whereas when it is a larger molecule, it is going to need the help of proteins. So we are going to look at what are the proteins that are needed for transporting things, and how does the concentration of those materials across the membrane define how they are transported. So the key thing here is that to remember is that when you have materials at a higher concentration on one side of the membrane and a lower concentration on the other side of the membrane, essentially you have created a concentration gradient of that material. Okay, so if you have more molecules, let's say I have more sodium ions on one side, less sodium ions on the other side, I have a concentration gradient of sodium ions across that membrane. And the natural law of things is that uh, molecules move down the concentration gradient from a place of high to a low concentration. But when you are moving things against the concentration gradient, you need energy. So think of moving a boulder up the hill. You're just rolling it up the hill. You're definitely going to expend energy. So you're moving against the concentration gradient. Right? So here's a, a long distance overview of the different types of transport across plasma membrane. So we can basically divide transport into passive transport and active transport. Passive transport is movement that does not require energy because things are moving down the concentration gradient. And active transport is when you need energy because you're moving against the concentration gradient. Passive transport can be divided into simple diffusion, where you don't need proteins. An example of this is movement of carbon dioxide or oxygen gases, and also diffusion of water. Water is going to have a little bit more difficulty as we discussed before, but it can still go through simple diffusion. The other type of diffusion, which also does not require energy but moving down the concentration gradient, is facilitated diffusion. And that is carried out by two types of proteins called channel proteins and carrier proteins. We're going to study facilitated diffusion in more detail in the next chapter, so I'm not going to discuss that more over here. Active transport can be divided into primary active transport and secondary active transport. And since you're transporting against the concentration gradient, you need energy, and that energy comes from ATP, adenosine triphosphate. So, if you remember from one of our previous studies, when you take ATP, the energy of the sun essentially is stored in the phosphate bonds, the um, adenosine triphosphate, the three phosphates, the energy between the first and the second and the second and the third. When you break that, that energy is used for powering the pumps, the protein pumps that are going to move things against the concentration gradient. So, um, essentially there are three types of proteins: channel proteins, carrier proteins, and pumps. Pumps, as we just saw, are required for active transport because you're pumping against. So here you see energy is being used, and that energy is used to move things against the concentration gradient. When you have um, uh, passive transport, that can be facilitated through channels, and carriers can facilitate passive as well as active transport. So we'll see the difference between carriers and channels. Think of channels as tubes, channel proteins as tubes, Okay, so tubes that span the entire breadth of the plasma membrane. So if you keep that pipe open, then things are able to flow through that tube. A carrier protein, on the other hand, physically carries things from one side to the other side. So the molecule that needs to be transported binds to the carrier protein, and the carrier protein undergoes a physical change in shape, and then it essentially transports it across the breadth of the membrane and then releases it on the other side. So that is the difference between a channel protein and a carrier protein. So coming back to active transport, as we saw, requires energy, and you can. So there are two types of active transport. One is called primary active transport, and the other one is called secondary active transport. When the movement is powered by hydrolysis of ATP directly, that is called as primary active transport. But when you are coupling the transport of one molecule with another molecule, moving along its electrochemical gradient, that is called secondary active transport. So I'll show you in a second what that means. So when you are in secondary active transport, basically what is happening is uh, you have ATP um, creating a change in the protein because of the energy it releases, and that causes the movement of ions from one side to the other side. And what that does is it creates an electrochemical gradient, meaning there is charge on one side and less charge on the other side. This change in charge essentially promotes the movement of other molecules that need to be transported across the membrane. So because this is not directly the second molecule is not moved directly because of the ATP, but because of the gradient, electrochemical gradient created by the movement of the first molecule. That's why we call it as secondary active transport. So here, if you have molecules moving in the same direction, for example, okay, we have ATP powering the movement of, let's say, sodium from one side of the membrane to the other side of the membrane. That electrochemical gradient that is created there enables the movement of glucose to move in the same direction as the sodium ions. Okay, so if movement of the co-transported molecule happening in the same direction is called as symport. Sim means same. But if you are in this case in the sodium potassium pump, which is one of the major pumps in brain activity and intramuscular junctions and so on, is ATP powers the movement of sodium, three sodium ions across the membrane, and that creates a gradient which causes potassium to move through the same pump but in the opposite direction. Because it is moving in the opposite direction, it is called as antipode. So let me take you back to this diagram here. So you can see active transport uses energy, and it can be either a direct hydrolysis of ATP which is running it, and only one molecule moves from one direction in one direction. That is primary active transport. And when you have the movement of one molecule creating an electrochemical gradient causing the movement of molecules alongside co-transporting, that's called a secondary active transport. So when the two molecules that are moving in the same direction, then it is symport. When one molecule moves in one direction and that causes molecules on the other side to move again in the opposite direction, that's called as antipode. So examples of symport and antipode. Sodium potassium pump, as we just saw, is an antipode. Okay, that's this one, sodium potassium pump. Sodium goes on one direction and then potassium comes down the other direction. And then an example of symport is glucose moving with along with sodium. That's right here. So sodium is moving, and in that electrochemical gradient allows glucose to move in that same direction as the sodium. So this is a little bit more descriptive look at the sodium potassium pump. ATP becoming ADP, and the breakdown of that last phosphate group releases energy, and the phosphate comes and binds to this sodium potassium pump, causing the binding of the third sodium molecule, sodium ion, and the pump basically carries it from one side to the other side against the concentration gradient, and that causes a physical change in the shape of the pump, so that potassium is able to bind, and then the potassium now is pumped out on the other side against its own concentration gradient. Okay, so far we looked at how small molecules and uh, somewhat large molecules are moved. But what if you have to move really, really large molecules, or if you have to move bacteria across membranes? A situation where you might need a bacteria to be moved across a membrane is when you are looking at immune responses, right? So your white blood cells are some of your white blood cells are designed to swallow bacteria that they encounter. So you have a solid particle here, a bacteria. Your plasma membrane basically uh, curves around it, basically creates a little bowl around it, a space around it, so as to encompass that bacteria, and then it pinches it inward so that that solid particle is now captured within a vesicle. Okay, so when you're taking a solid particle like this is called as phagocytosis, which means um, uh, eating a cell here, cell eating. But you can also use the same kind of infolding, membrane infolding, to capture liquids and then bring them into the cell. So that is called as pinocytosis, because cell drinking. Okay, there's also another type where you have receptors. Receptors are proteins that can bind to specific uh, proteins. So you can have receptors in on the surface of the plasma membrane, and those receptors bind to the proteins that they are supposed to bind to, and then the whole thing kind of um, curves inward, and there is a specific kind of protein coated here called clathrin. So anyway, this clathrin coated pit basically engulfs this molecule, and then um, and then takes it in as a vesicle. So that's called receptor mediated endocytosis. So endocytosis, essentially what you need to remember is it's taking in large particles, and it happens because of infolding of membranes, and it forms vesicles in the process. If it eats, if the infolding encompasses a solid particle, we call it cell eating or phagocytosis. When it encompasses a liquid being taken in, it's called pinocytosis. When that process, when the taking in of solid particles uses receptors on the surface of the membrane, we call it receptor mediated endocytosis. And finally, we are looking at the opposite of endocytosis, which is exocytosis. Exocytosis is where you have molecules being moved from inside to the outside. So it could be because your molecules are being secreted out, or you have, let's say, processed a bacteria and that cell wants to spit out all the unnecessary material. Either way, the cell forms a little vesicle with the stuff that it needs to spit out, and then it takes it to the membrane, and this membrane here, the vesicle of the membrane of the vesicle fuses with the plasma membrane, and releases whatever it needs to release to the outside space. That's called exocytosis. Okay, so here's a very quick review in summary. Simple diffusion involves movement of small particles from high to low concentrations across the membrane. Small uncharged non-polar molecules. When you have slightly larger molecules or ions at higher concentration on one side, and you're moving down the concentration gradient, you can use either channels, which are just tubes, or carriers that are going to physically carry them by binding to them and moving them to the other side. Diffusion and facilitated diffusion, simple diffusion and facilitated diffusion are all part of passive diffusion. Active diffusion, or I'm sorry, active transport requires energy, and that is carried out through pumps. When the direct hydrolysis of ATP to ADP powers the pumps, then it's called as primary transport, primary active transport. When the transport of materials, let's say here we have the green particle here, the movement of the green particle is powered by the movement of another ion, which again requires energy to move. So that power creates a concentration gradient which allows this molecule to go from one side to the other side against the concentration gradient. Then we call it as symport if it happens in the same direction, and antiport if it happens in the opposite direction. So I know there was a bunch of new terms. Hopefully you got the design of how all active and passive transport happens, and how the different types of um, diffusion and active transport are categorized, and what proteins allow them to move across the membrane. Okay, thank you very much.